Everybody okay? Everybody can hear me? My name is Ryan Campbell. I'm not full screen. I thought I did. Hi, my name is Ryan Campbell, and I want to talk to you about a project that's ongoing in the Yoder Lab at Duke University, where we're going to try to directly estimate the germline mutation rate of mouse lemurs, or the genus Microcebus. So I probably shouldn't have to tell everyone in the room how important mutation rate is, but as it always goes, when someone says that in a talk, I'm still going to. And for instance, mutation rates apply to all the raw material for evolution. I'll apply some of the raw material for evolution. And if you kind of cast your mind back to any talk you've been at today or yesterday, you can probably see where mutation has an effect in that talk. However, we often measure mutation rate indirectly. We don't measure it, from a trio. We measure it at a population level. We also know things about evolution such that it's correlated with effective population size, generation time, and body size. Another question, I guess, is why am I working in mouse lemurs, or why do we care about mutation rate of mouse lemurs? Besides the fact that they're adorably cute, they are actually a really good species to test these drivers of mammalian mutation rate. They're a primate that also has a high effective population size. And as I go into more depth about mutation rate, it's kind of an area of the tree that has been unplumbed or kind of unstudied in this area. And the other reason, and perhaps probably more pressing to our lab, is that it'll improve the understanding of the evolutionary history of Madagascar. So in our lab, we look at the evolutionary history of this group, and we build a tree. And when we build that tree, we estimate the mutation rate, or we tell the whatever program we're using, the mutation rate somewhere. It falls somewhere between mice and humans. And if that's off or if that's wrong, that could drastically throw our inferences out of whack in time. So what we do know about the history of Madagascar and this genus is that it's about an 8 million year old radiation with roughly 20 or a few more species. However, some of these species are very young. Some are about 55,000 years old. And like I said, if that mutation rate we're using to establish that a species of 55,000 years old was off, then we could push that number higher or lower, and it would really change some of the inferences that we're making, not only about the species age, but about the rates of speciation in the genus, and as well as the ecological history of Madagascar. The things we know about mammalian mutation rate mainly come from model organisms. Things like humans and chimps and mice are the main systems that we've done trio studies in of mutation rate. We've been able to establish the mutation rate of these organisms, also the spectrum of the mutation rate, so which bases are changing to which and how frequently. And then often the studies go into looking at where these mutation sites happen, because that's pretty important as to what parts of the genome have higher or lower relative mutation rates. And so looking across mammals, we see that effective population size has a negative relationship with mutation rate. You've got humans here, and the width is sort of a wide range of estimated effective population sizes throughout the literature. And then vertically we have the mutation rate, and so you can see that as that effective population size grows, it's higher in mice, and they have a lower mutation rate. Similarly, looking at the chimpanzee studies, you can see that CPG islands are enriched for mutations, and C to T mutations are also very common. And so basically what I like to be doing here is situating mouse lemurs within these findings, asking the question, do mouse lemurs follow their effective population size? Are they more like primates? And kind of learning more about them. So for methods, we used the Duke Lemur Center and found a pedigree of 2 parents and 2 offspring. So it's 2 trios technically. We used 10x genomic sequencing. Their assembly software is called Long Ranger. VARScan was used to pick, to identify the number of mutations, and finally place them in the genome. So I'll go through that, each of those in a little more depth. We had 2 parents, Poblano and Sweet Annie, and then they had 2 focal offspring that were born in different years. So there was 2 different breedings from different years of this pair. And we also sequenced Poblano, the sire, twice, and that'll come in handy for the analysis. Basically, we wanted to make sure you just kind of have like a double check, so we could use everything we did with Poblano A version and Poblano B version. The sequencing, we used 10x genomics. I'm not sure how many of you are familiar, so I'm going to do a quick, and dirty discussion of what 10x genomics is. Basically, it's a pre-fragmentation barcoding process that works within the Illumina pipeline. When you have nice long pieces of DNA on the order of megabases, You encapsulate them and barcode them uniquely with about 300 or 400 barcodes. And then you go on and fragment those pieces, and the first step in assembly is to kind of reunite all the pieces that originated together on the same strand, right? So when you're assembling, you actually are moving beyond the small windows of a usual high-seq library that's 300 or 400 base pairs and looking in the order of hundreds and thousands and megabases of haplotype assemblies, which is actually really useful if you're looking for mutation and parent of origin effects and things like that. And then, aside from that small little side step, you go on and sequence at 30x depth on an Illuminati sequence. So for assembly and mapping, we assembled the reads with Long Ranger, which is the 10x program. They go in and look for, like I said, all the similar barcodes, group them all together, and you end up with some nice long haplotypes. I'll show you the results in a minute. And then we also have a Microcebus morinus genome. It's 3.0 now, and so it's chromosome level, and we were able to map one of these longer haplotype reads back to that genome. I use VARSCAN to identify new mutations. VARSCAN software that basically takes BAM files and looks from two parents to an offspring and looks for places where there's a novel mutation in the offspring, but also enough coverage in the parents to confirm that there was not a reference, that the call was not just non-reference, but there was depth in all three individuals. And lastly, I took a number of steps to double check these mutations. This was actually one of the trickier parts of the work, but I wanted to exclude mutations that were found in any other individuals in our colony. So looking at across other data sets, not just from this trio, but elsewhere. So you can imagine a case where you might think an individual has a genetic mutation, but somewhere else in the pedigree or somewhere else in the same colony it exists. Maybe it's just missed in the parent, and that's a way to check for that. We also compared Long Ranger results with the GATK alignment, just to kind of have a secondary check. I wanted to make sure that there wasn't something funny about Long Ranger, and just to have that backup is good. And then, like I said, we sequenced the mono to sire twice, and so we compared across both versions of the father to make sure that it was present in each. So, as for results, we had, like I said, 4 individuals, 5 total samples, because we sequenced the one twice. We ended up with about 34x coverage on average across the 5 samples, and our N50 just from this assembly was about 1.2 megabases. I think the 10x software, or the 10, I was somewhat skeptical going into 10x, but it's been a fairly smooth process, and the data coming out are pretty amazing. So if you're anywhere away from a genome, having these long haplotypes is really, really useful. And so I was very impressed with the amount of coverage and the amount of data that added, and the layers that could add to our study. We found 81 total de novo mutations across the 2 individuals. You can see them labeled here and there. And that gave us a mutation rate of somewhere around 8.8 times 10 to the negative 9 mutations per generation. So that's just a number with a large exponent or a small exponent. Where does that go in the graph that we showed of other mammals? And it turns out it fits in right about here. Pretty much what we would expect. So it's basically being somewhat affected, perhaps, by its affected population size. It's definitely not in with other primates in terms of vertical mutation rate. And this is actually a good news, because in our lab, like I said, we estimate with a Bayesian prior, it runs from human to mouse. So while it's not going to drastically throw off any of our tree results. And so beyond just the rate, I wanted to look at a little more about where in the genome these mutations were happening. So what we have in front of us here is a histogram, in essence. The darker, allegedly blue part of the histogram is mutations that were happening within a gene, so between the start and the stop position of a gene. And then this is upstream, or this is downstream, and this is upstream. Just plotting mutation to the distance to its closest gene, and you can see that some happen in genes, some are before, some are after. This actually matches. I was actually surprised to see that about half ended up within genes between start and stops, but looking at the genome annotation that we have, about half of our genome is between start and stops of genes. So not terribly biased as to where they're falling. As for the spectrum, you can see that we have the same kind of pattern here of C and T. So there's a lot more C to T mutations, kind of like we saw in the other mammals, which is good. In fact, they're right here, just above chimp, but below mouse and human. So that's, evidence that there's not this drastic changing. There's nothing different, nothing terribly different about mouse lemurs or lemurs that from the other mutation patterns in mammals. And one thing I wanted to try to address last is looking at the mutation rate of CBG islands. And I have islands in quotation marks because there hasn't been any epigenetic studies of mouse lemurs or lemurs at all. So when going about tagging an area as a CBG island, I had to refer to what ended up as CG-rich islands, right, or CG-rich areas of the genome. On the left, this is the overall, violin plot, sideways histogram of genome-wide CG percentage. And on the right is the CG percentage of our mutations that I counted. So 200 base pair window around the mutation, and what is the CG content of those areas. So the genome-wide mutation rate is here, and you can see that the mutations do have a little bit of a higher end, a little bit of a higher GC content around them. I also wanted to kind of compare. Once I decided to make a cutoff at about 55%, which is the top quartile of the genome. That also kind of jives with what other mammals have seen as CBG islands. And so once I labeled this top 25th percentile of the genome as a CBG island, in quotes, looking at the mutations that then also fall in that, where do they fall out along this spectrum? Or do we see them in the same pattern as we saw other mammals? And it turns out that it actually behaves really well. You see that there's a lot of, a high proportion of these C to T mutations are happening in CG-rich areas. Lastly, I wanted to see if it wasn't just that the CG-rich areas had C to T mutations because they have Cs in them. And so I tried to plot, or I applied this is the same gene start position on the x-axis here, looking at the density of where these islands are. And basically the blue line is the CG-rich areas, and you see that they are kind of near the starts of genes, which is what we would expect if they are active or they are actually CPG islands. So I think that's, like I said, we haven't done any methylation work, but I think it's pretty fascinating that they're kind of aligning with the right areas of the genome. All right, so in conclusion, we are in line with predictions from NE, from other mammals, and the spectrum resembles other mammals, and the islands also kind of resemble other mammals. In terms of evolutionary history of Madagascar, our mutation rate that we had been using prior was accurate. It reduces, this mutation, knowing this mutation will reduce the width of the prior we were having to use, and even further work with this to establish a solid confidence interval will make those estimates or those inferences from trees even more accurate. So, mouse lemurs conform with the current theory of NE and mutation rate. Their pattern resembles other mammals, and their prior assumptions of our work have been accurate and will be further improved. I'd like to thank the funding sources, the NSF, Duke Biology, and the Lemur Center for all the samples, and our lab, Kelsey, Peter, and Rachel. That was fun. Lastly, we have a poster tonight at 6:30. Kelsey is giving a poster in the poster session, and tomorrow, Love Lemurs, Dwarf Lemur, Evolutionary Histories at 10:15. Thank you.
To estimate the mutation rate, how did you estimate the denominator?
So I threw out, mainly looking at sites we had enough coverage, and then I threw out the X chromosome because I kind of ignored the X chromosome. I'm looking, so one of the software I was going to use was Genomics, but it wasn't working like that. It wasn't, yeah. You fixed it, I think. I got an email last week from GitHub. It's like, it was fixed. So yeah, I want to use Genova here once I think the output from Long Ranger wasn't perfect, so it was going to
I think you could use a data set like this to estimate the rate of other genomic changes, like insertion and deletion.
Okay. I think
I feel like that's sort of like
I would be concerned that they would be even more rare. Right?
Yeah.
Like so your numbers
That sounds like more so.
Yeah, more samples, and it is not a natural population, unfortunately. It's a pedigree population. But yeah, it would have the same, maybe even more problems of like inference in terms of saying like, oh, I see 2 insertions, but we're definitely looking at that, and the haplotype data makes it really